Hello students, welcome to Mahesh Science YouTube channel. We are discussing chapter cell cycle and cell division. In the last video, we discussed it regarding meiosis 1, particularly in that prophase 1. That is, prophase 1 is having 5 subphases, that is leptotene, zygotene, pectine, diplotin and dikinesis. I hope you have seen that video. Okay, if you have not seen that video yet, okay, now you visit to that video, then come back to this video. Here we are going to talk regarding metaphase 1 and next stages of the meiosis. Okay, here in meiosis 1 we are going to right now talk what metaphase 1. Okay, in during prophase 1 5 subphases took place. Okay, under the last phase, that is dikinesis. See here what changes occurred during the dikinesis in the prophase 1. Let us see. See here the centrosomes started moving towards the opposite poles. Okay, like this. Then nuclear membrane got disappeared. Nuclear envelope got disappeared during the dikinesis, and chiasmata get separated, meiotic spindle formed, all other cell organelles, okay, all other cell organelles were formed, disappeared, okay, all these things took place in the dikinesis. During metaphase, what happens? See here, the nuclear envelope completely disappears. Nuclear envelope completely disappears during the metaphase. That marks the beginning of the metaphase, and the centrosomes are exactly at the opposite poles. Centrosomes are exactly at the opposite poles, okay, with the formation of what? Spindle fibers. This is how they formed the spindle fiber. And centrosome, chromosome, particularly tetrad now comes on the central equatorial plate. Okay, during metaphase 1 already homologous chromosome paired and formed a tetrad or bivalent. Okay, those tetrad or bivalent chromosomes come and lie on the central equatorial plate. Okay, this is how a tetrad comes and lies on the central equatorial plate. Observe here one important change. Okay, during mitosis, what we studied during mitosis, see here all the chromosomes, okay, whatever, how many chromosomes are there, they all come on the central equatorial plate. Okay, see the difference between mitosis and meiosis. See here, in mitosis all the chromosomes come and lie on the central equatorial plate, and spindle fiber attached to the kinetochore of the centromere of the chromosome. Okay, this is how they aligned in mitosis, that is metaphase of the mitosis. But now all the chromosomes not aligned in the central equatorial plate. Instead, a tetrad or bivalent formed by the homologous chromosomes that came on the central equatorial plate. And spindle fiber attached to the centromere of the homologous chromosome. Not a sister chromatids. See here spindle fiber got attached to what? Kinetochore of the centromere of a sister chromatids. But here spindle fiber got attached to the kinetochore of the homologous chromosome. That was the big difference here. This is what metaphase 1. Then in the next phase, anaphase, what happens? Let us see. In anaphase 1, what happens? See here. The homologous chromosome gets separate. See here in anaphase, centromere is not getting dividing as like that of the mitosis. In case of mitosis, what happened? Centromere splits and half of the sister chromatid move towards the one pole and half of the sister chromatid move towards the opposite pole. During what? Mitosis, especially anaphase of the mitosis. Here centromere is not splitting. Instead, half of the chromosomes are moving towards the one pole and half of the chromosomes are moving towards the opposite pole. One of the homologous chromosomes, okay, one of the homologous chromosomes move towards the one pole. Let's say a red colored chromosomes move towards the one pole and blue colored chromosomes move towards the opposite pole. This is how the anaphase appears. See here, I am going to make a changes in the same cell itself. See here, the shell shape slightly changes in the anaphase, and half of the chromosomes are moving towards the one pole and half of the homologous chromosomes are moving towards the opposite pole. That's why the chromosome number reduces to half. This is what the reason, because chromosome number is getting reduces to half. See here, if the parent cell is 2n, here in the anaphase, half of the chromosome will move towards one pole and half of the chromosome will move towards the opposite pole. This is what the reason chromosome number reduces to half. That is why meiosis 1 is the reductional division. Meiosis 1 is the reductional division. Remember, there is no splitting of centromere. Okay, no splitting of centromere. Instead, the whole homologous chromosome among all the four, let's say there are 4 chromosomes. 2 chromosomes are moving towards the one pole and 2 chromosomes are moving towards the opposite pole. Here, sister chromatids not got separated. Still, sister chromatids are there on the homologous chromosome. Note this point. It is going to be very, very important. Here, sister chromatids not got separated. Still, they are on the homologous chromosomes itself. Okay, half the number of chromosomes move towards the one pole and half the number of chromosomes move towards the opposite pole. That is why the ploidy of the cell changes by diploid to haploid. Okay, this is what anaphase 1. Moving to the next one, that is telophase. Telophase 1. In case of telophase, let's see what happens. All the changes that occurred during prophase will get reversed here. All the changes that occurred during the prophase will get reversed here. Nuclear envelope reappears. Okay, nuclear envelope reappears. Nucleolus reappears. Then chromosome will decondense and become into what? Chromatin. Chromosome will decondense and become into what? Chromatin, and the shape of cell turns to almost 8 shaped. 8. Okay, this is what the changes occur during the telophase. And telophase 1 is followed by cytokinesis, in which equal distribution of cytoplasm to the 2 daughter cells will happen, and a furrow in the plasma membrane will form, and which get deepens from external to inside, so that the daughter cells will get separate by the end of the meiosis 1. Now tell me, by the end of meiosis 1, how many daughter cells are formed? See here, 2 daughter cells are formed. What is the ploidy? They are haploid in nature. two haploid daughter cells are formed by the end of meiosis 1. I hope all the points regarding metaphase 1, anaphase 1, telophase 1 and cytokinesis are clear to you. Now let's move on to the next phase, important phase, that is meiosis 2. Okay, meiosis 2. Even this meiosis 2 is further divided into 4 phases. Okay, let's understand them with the help of diagrams. Meiosis 2. Meiosis 2. Okay, in meiosis 2 also there are 4 phases: prophase 2, metaphase 2, anaphase 2 and telophase 2, just same as the meiosis 1. But by the end of meiosis 1, see here, by the end of meiosis 1, two daughter cells are formed. Okay, two daughter cells are formed. They are something like this. Okay, two daughter cells are formed and they are haploid in nature. Okay, now we are talking about meiosis 2. Meiosis 2. In between meiosis 1 and meiosis 2, there is a very thin marginal phase is there. Okay, there is a thin marginal phase called interkinesis. Interkinesis. Okay, see here, by the end of meiosis 1, how many cells are formed? Two daughter cells are formed. Okay, and this stage is also called as dyad. Okay, dyad. Dyad. Okay, after the formation of dyad, okay, by the end of meiosis 1, it enters into what? Interkinesis. It is of very short duration. Then it moved to the meiosis 2. In meiosis 2, it first enters into prophase 2. Let's see what changes occurs in the cell during prophase 2. Let's assume here whole meiosis 2 can be explained in a single line. What was that single line? Here, if you remember that line, you can easily write all the phases of meiosis 2. Meiosis 2 is just same as the mitosis. That's it. Meiosis 2 is that just same as that of mitosis. That means all the changes that occurred in mitosis will all get happen here. Okay, all that get repeat here. Okay, see here. Here in meiosis 2, all the changes will same as that of the what? Mitosis. Let's see what happened during prophase of mitosis. See here, the nuclear membrane gradually disappears. Okay, nuclear membrane disappears. The chromatin will get start condensing and becoming into chromosome. Okay, chromatin will get start condensing and becoming into what? Chromosome. Okay, in both the cells the similar changes will occur. Okay, nuclear membrane disappears, chromatin will get condensed and become into what? Chromosome. Okay, and nucleolus disappears, other cell organelles disappears, the what? Centrosome move towards the opposite pole with the formation of a spindle fiber. Okay, if you remember mitosis and their all phases, you can easily write meiosis 2. Okay, this is what? Prophase 2. Then in metaphase 2, let's see what happens. In metaphase 2, yes, all the chromosomes come and lie on the central equatorial plates. Okay, okay, this is how the metaphase is there. All the these chromosome will come and lie on the central equatorial plates like this. The same as prophase. Okay, then in spindle fiber attaches to what? Centromere. centromere on a disc shaped structure is called kinetochores. Okay, this is what metaphase 2. Then in anaphase 2, see here, anaphase 2, what changes occurs? Okay, see, the centromere splits and chromosome will move towards the opposite pole. Centromere splits and chromosomes move towards the opposite pole. Okay, now the sister chromatid splits, remember. Now what happens in anaphase 2? Sister chromatids will get separate from each other and move towards the opposite pole. Okay, these changes will occur in both the daughter cells. Yes. This is how the sister chromatids move towards the opposite pole. By the end of anaphase 2, they almost reach the opposite poles. Opposite poles. And the last phase, that is telophase 2. Telophase 2. See here, in telophase 2, okay, all the changes that occurred in prophase will reverse. Okay, reverse, that means nuclear membrane reappears. Nuclear membrane reappears. Nucleolus reappears. Okay, then chromosome will decondense and become into a thin chromatin. Okay, like this. Chromosome decondense and become thin chromatin. Okay, this happens during the what? Telophase 2. And telophase 2 is lastly followed by cytokinesis. Telophase 2 is lastly followed by what? Cytokinesis. Here the one small furrow occurs in the plasma membrane, which get deepens from external to inside, so that uh, so that the cytoplasm is equally distributed to the both daughter cells. Okay, both daughter cells, like this. Yes, see, this is how the daughter cells will get separate by the end of cytokinesis. Daughter cells get separated. Hmm? Yes, now tell me, how many daughter cells are formed by the end of meiosis 2? Yes, tell me, how many daughter cells are formed by the end of the meiosis 2? Yes, you are correct. Four daughter cells are formed. Four daughter cells are formed. What is the ploidy? What is the ploidy of those daughter cells? Yes, once again you are correct. That is, they are all haploid. Haploid, haploid, haploid. That means four haploid daughter cells are formed. Now tell me whether they are identical or non-identical. Think over it. See here, whether the daughter cells formed are identical or non-identical. Remember, they are non-identical. They are non-identical. See here, by the end of mitosis, two daughter cells are formed. They are exactly same as the parent cell. They are identical daughter cells. But here, whatever daughter cells are formed, they are non-identical, because of the crossing over in prophase 1, the genetic material of all four cells got changed. That is why they are genetically different. Okay, non-identical daughter cells. Each and every gamete is entirely different. Okay, that is why no two siblings are exactly same. No two siblings are exactly same. just because of the crossing over. This is how meiosis brings about genetic variation, actually, which favors the evolution. That is what one of the significance of the meiosis. Okay, so here we studied one significance of meiosis, that is, it brings about genetic variations, and that genetic variation supports evolution. Then what is the other significance? Think over it. Yes, meiosis helps to maintain constant chromosome number to a particular species. That is what the another significance. There are two significance of meiosis. Okay, let me repeat. The first significance is it brings about genetic variations which supports the evolution. Okay, the second and most important significance of meiosis is it helps to maintain the constant chromosome number for a particular species. Okay, with this we completed meiosis 2. Okay, now can you tell me what's the difference between mitosis and meiosis? Okay, but with that we can sum up this whole chapter. Okay, yes, tell me what is mitosis? Mitosis is equational division, whereas meiosis is the reductional division. Here in mitosis, the same number of chromosomes will be there in the daughter cells. Okay, that's why it's a equational division. Here chromosome number reduces by half. That is why it's the reductional division. Okay, mitosis can takes place both in diploid cells as well as haploid cells. But remember, meiosis always takes place in diploid cells. Okay, it never takes place in haploid cells. And then mitosis takes place in somatic cells, means bodily related cells, whereas meiosis takes place in germ cells or sex cells, or you can say gamete mother cells, myocytes, like that. Okay, then by the end of mitosis, two daughter cells are formed. Okay, by the end of meiosis, four daughter cells are formed. And in case of mitosis, daughter cells formed are identical to each other. Okay, whereas in meiosis, daughter cells are formed are non-identical to each other. Okay, yes, in mitosis, okay, in mitosis, which helps for organisms growth, okay, and it also favors the cell repair, whereas meiosis helps to maintain constant chromosome number in that particular species, as well as it brings about genetic variations. Okay, mitosis results into asexual reproduction, whereas meiosis results into what? Sexual reproduction. In case of mitosis, there are only four phases: prophase, metaphase, anaphase, and telophase. Whereas in meiosis, how many phases are there? Okay, it is divided into meiosis 1 and meiosis 2, each having four subphases: prophase 1, metaphase 1, anaphase 1, telophase 1. Like that, prophase 2, metaphase 2, anaphase 2, telophase 2. Like this, here 8 phases are there. Here in mitosis, 4 phases are there. Differences. One more difference I am recalling right now. Okay, that you have to compulsory mention. Okay, here in case of mitosis, there were no pairing of homologous chromosome and crossing over. Okay, there were no pairing of homologous chromosome. Whereas in case of meiosis, there is a pairing of homologous chromosome. And in mitosis, there were no crossing over. Whereas in meiosis, there is a crossing over. Okay, like this. If you find still more differences, then just comment in the comment box. If you like this video, please hit a like button, get subscribed to my channel, and share it to your friends. Thank you so much.